Good evening, everyone. I would like, as always, begin by acknowledging the land that we're on, which is the traditional land of the Dakota people. Welcome to the Weisman Art Museum and to the guest lecture by Oron Catts. I'm Boris Orkerman. I'm a curator for collaboration in this museum. Sorry? I am Cindy and Jay Allenfeld, curator for creative collaboration. I will acknowledge them, I promise. It's difficult to work with the boss in the front row, I don't know. This is a two-part event. There will be the lecture, and then Brenda Ogley, Mark Borrella, Steve Kelly, and myself will join Oron on the stage for a panel. Both parts are open for questions at any time, so if you feel like asking a question, please raise a hand. Just please wait for the microphone to arrive to you. This event is being recorded. This lecture is part of Oron's week-long visit to the U. That visit was made possible by a collective effort. Thank you so much to Michael Winnikoff of the Biotechnology Institute for co-organizing this visit with me. I would like to thank the following people and organizations for their financial support. Thank you to the voters of Minnesota for supporting the operational budget of this museum through the Minnesota State's Art Board. Thank you to the Wells Fargo Foundation Minnesota also for supporting our operational budget. Thank you to the Biotechnology Institute, the Medical School, the Institute for Advanced Studies, Minnesota Discovery Research and Innovation Economy Partnership, School of Architecture, Interdisciplinary Center for the Study of Global Change, Department of Art History, College of Biological Sciences, and the Bell Museum for their financial support of this visit. Thank you to the Target Foundation for building the studio that I curate. Thank you to Cindy and Jay Illenfeld for the endowment that makes my position here possible. And huge thank you to my colleagues at the Weisman Art Museum for supporting this visit and for making this event happen. And of course, thank you to Oron for coming all the way from Australia. If you read on the history of contemporary art, you'll see expressions such as art and life quite a lot. If you Google the expression in Google search, you'll find, I think, about 1.5 million hits. The usual context for those will be the proposition that the modern Western art has evolved to be created, experienced, and discussed in isolation from the life routine. Artists create art in isolation of studios, critics analyze works of art as isolated acts of creativity, and public disconnects from the mundane and enters museums such as this one to enjoy art. Many innovations in art were driven by artists who were not happy with this isolation, such as Predvizhniki in 19th century Russia rebelled against the academy and went to work in a rural country, or the happenings and the conceptual art of 60s and 70s that moved to the public space, or the socially engaged artists of today who directly address social issues. All that was with the agenda to reconnect art and life. The word life in all those reconnections is really more of a metaphor. It stands for things that happen outside of art. What that word usually does not imply is life in its literal biological sense. The need for this literal connection became apparent with the advance of biotechnologies and with the massive migration of modified life products from laboratories to our daily routine through food, medical care, and media. With this process came the need for an informed and critical discussion of life sciences and for art that provokes and maintains this discussion. Oron Catts is one of the artists who took this art-life connection literally. Over more than 2 decades, his work is concerned with bringing artistic critical approaches to the lab and the awareness and critique of the cutting-edge life sciences to general public. One of the core questions in this art-life connection has always been, what is the actual effect we as artists have outside of the gallery? How do we know whether our art actually really has consequences? Oron's career happened in very unique times where public perception of life sciences saw major shifts. We might know very little about biology, but most of us will remember the sheep dolly, see the GMO or non-GMO labels on groceries, and hear about stem cells controversies. It can be claimed that the art of Oron Catts and other artists and collectives such as Stellark, Eduardo Katz, and Critical Art Ensemble not only happened to be at the same time as those shifts, but actually had a direct impact on them. This is one of the claims that Oron will develop in today's lecture. Oron's artistic focus in biological life is unique on itself, but there is another aspect that makes his work really stand out, especially in the context of the Target Studio collaboration program that I curate. This program supports working collaborations, working relationships between artists and researchers at the university. One of the biggest challenges in my work is sustaining those relationships. How can an institution such as this university support collaborative work in the long term, beyond fixed-term projects with predetermined goals and timescales? Oron's work with the Department of Human Biology at the University of Western Australia began in 1996. In 2000, he and his partner Yonad Zur have established the Symbiotica, the artistic laboratory within the same department. Remarkably, Symbiotica remains operational ever since, creating art, developing original research, teaching academic programs, hosting residencies, curating events and exhibitions, publishing books, and more. In my knowledge, this is an unprecedented achievement, a sustained artistic production that is embedded within non-art academic department for such a long period of time. In the lecture today, I hope we will learn something about how that happened. On my end, I'm really interested to learn how we can do something of a kind here at the university. So before I hand the stage to Oron, here is a call. If you're interested in the same thing, talk to me. We'll have a better chance to figure it out together. And also, there are still seats available on 2 workshops that Oron will run this week. Biotechnology for artists and DIY biology for scientists. If you want to take part in those, talk to me. And without further ado, the stage is yours.
Yeah, thank you so much, Boris, and thanks everyone else who was involved in bringing me over here. I won't repeat it, I won't remember. I would also like to acknowledge my lab, as Boris mentioned, is in Western Australia, in Perth, on the shores of the Swan River, which is the Whadjuk people of the Noongar Nation land, and I would like to pay my respect to their elders, past, present, and emerging, and particularly thanking them for allowing me to do so much mischief in their land and still be around. A couple of trigger warnings. I know it's compulsory here, mandatory. There might be some images that you would find disturbing. So I don't know if I'll let you know before, but just be aware that there might be some blood and gore because this is live. Also, my talk is going to move into areas where we don't have language to describe, and I tend to swear when that happens. And I find that swearing is actually one of the most expressive ways of engaging with the lack of language. So just apologies if that's going to happen. I'll try to avoid it, but if it comes, it comes. So I would like to start with a statement from a recent publication from the World Economic Forum that tells us that they face the task of understanding and governing. We face the task of living in a world where we have 21st century technologies that are engaged from 20th century's mindsets and 19th century's institutions. Institutional change is therefore critical to overcoming those challenges, but so is the mindset, which is really what I'm interested in, adapted to the 21st century challenges we face. And in other words, I don't really agree with a lot of what Yuval Harari says, but I agree with this statement that he says that we inherited our current political systems, whether communism or liberal democracies, from the Industrial Revolution, and he doesn't think, and I agree with him, that either of them can survive the completely different realities of biotechnology and artificial intelligence. And I think that we're already starting to see that things are falling apart. Yeah, so I don't want to go into American politics, but you can get the drift. I won't go into British politics, but all of those are signs towards people who are striving to find some connection with this disconnect between the 19th century institutions, the 20th century mindsets, and the 21st century challenges that we're facing. And in my perspective, because I'm interested with life, It's really about kind of this really strange situation. We're in a perfect storm at the moment where technology becoming more and more lifelike. Yeah? So we give technology autonomy. We already kind of, if you want to talk about it in terms of psychopathologies of control, we already relinquished our control over human-made systems. Yeah? So think about our economies, think about algorithms that even the programmers have no understanding how they operate, think about driverless cars, think about killer drones. Think about life and death decisions that we already delegated or relegated to our technologies without us having any say in it. At the very same time that our psychopathology of control drive us towards trying to control systems that existed outside of our control for ions and that's living systems. Yeah? So things like synthetic biology, this idea where engineers are talking about rationally designing life and creating factories in cells and talking about all of those things at the very same time that we already acknowledge that systems are so complex that we have no control over them. So there's this disconnect again, which can create some anxiety with some people. So, but it's kind of, it's not very new. This is something I really like to show. This is a pork packing factory in Cincinnati. I'm sure around here there are many of those as well, around the mid-19th century. And the story goes that actually Henry Ford went to see those disassembly lines where complex systems were broken down into abstract components, and that's how he came up with the assembly line itself. Yeah? So life and industry are always connected, and we tend to kind of go through cycles where we are kind of using life both as a metaphor and as industry and vice versa. And one of the very first people to kind of to talk about it in terms, and this is I hope Mark would pick up on in our conversation later, is Jacques Laub, the German-American scientist who in the early 20th century was already talking about the idea of biology becoming an engineering pursuit. Yeah? So moving away from a merely analytical, from a merely descriptive discipline to a prescriptive discipline where we can start to engineer life, and life can become a raw material for our needs and wants. Another scientist that was working around the time is Alexis Carrel, and the term artificial life was actually coined to describe the work of Laub and Carrel. Carrel was a French scientist that won the Nobel Prize for Medicine in 1912, came and established the laboratory of experimental surgery at the Rockefeller Institute, and was the person who was known as the person who developed the whole field of tissue culture, the abstraction, the taking out of parts of complex organisms and the maintenance of those parts outside of the original body from which they were taken. He's also known for many other things, including writing a pseudo-philosophical book called Men the Unknown in 1935, which among other things also recommended the use of gas chambers to eliminate undesirable elements in human society. He was a surgeon, yeah, he was a doctor, and that's doing good in the world. He also collaborated with a local hero, with Charles Lindbergh, on the organ perfusion pump, and actually, as far as I can understand, the story of them working together spread rumors about him creating an iron heart for Charles Lindbergh and creating actually the original Iron Man. Yeah? So popular culture kind of evolved from those kind of works as well. But in the context of Carrel, I want to talk to you about kind of the realization that he and his colleagues, this is one of his colleagues stating in 1916 that through the discovery of tissue culture, they, so to speak, created a new type of body in which to grow the cells. So this combination between life and technology became very, very intimate, where the technology becomes the body. Now, what I would argue is that we are still lacking a cultural understanding to be able to cope with those things, that those things are sitting in some strange zone where we can't really articulate them, and I would demonstrate it through this. This is the very first premature baby board here in the United States. It was part of the Pan American Expo in Buffalo in 1901. It wasn't part of the technology section, it was in the oddity section. And when the Pan American Expo shut down through to, what was the name of the president that was murdered there? Yeah, first assassination? Yeah, anyway, there was a murder of one of your presidents, many others to come. They moved the whole operation at 1901 to Coney Island. And that was one of the very first permanent installation in Coney Island. And you might be able to see there, it's infant incubators and living infants. They were selling tickets. That was a freak show. And it lasted for about 30 years. There's many, many reasons why babies in incubators ended up as freak shows. I would argue part of it, so there are obviously eugenics, There are issues around, because those were broken babies that no one wanted, so there's no reason for them to live unless they can make money. The other reason was that obviously childbirth at the time was women's business. It wasn't kind of technologicized, so there's no place between women's business and technology, so it's only a freak show. Those babies obviously were very unique. They were the tiniest humans available for viewing. And I would argue the main reason was that there was no other cultural place to put them in. Yeah, so there was no way to articulate the existence of human bodies inside machines, and the only place they could end up was as freaks. And this is coming again. The new biology creates new types of freaks that we as a society, we as a culture, have no tools to engage with. And as an artist, this is exactly what I'm interested in doing. And just again, to put it in context, even when the medical establishment was interested in those, because they had 80% success rate, and they started to distribute them to hospital and amusement park from Dreamland, Coney Island. Yeah. Now we jump across to Manhattan. As far as we know, the first time that an artist actually displayed manipulated living matter as in a context of art, obviously there's many other, you can think about flower arrangements, you can think about bonsai, but we think of the Western artistic tradition. The first time that we can kind of trace it back is to the work of Steichen in 1936, where he accompanied one of his photographic exhibitions in MoMA in New York with a set of flowers that he manipulated by introducing those delphiniums to different types of mutagenic agents. And this is kind of always kind of a point of reference for us. For me, one of the most important images of the late 20th century was actually done by scientists. So as an artist, I was really interested in this connection, as Boris said, between art and life. And then 1995, this image hits the media. This is the surrealist dream comes alive. This is kind of our dreams that humans always imagined, this animal-human chimera, and here it is jumping on the new stage, which is the Petri dish. And the Petri dish would appear time and time again within this context as a new stage, the new theater of life, where life is being constructed, manipulated, engineered, and sculpted. Yeah? So this is the BBC image from the 1995 documentary. Obviously, it's a stage image. This is the image from the actual scientific paper that the Vacanti brothers and their collaborators published in 1997. You can see, obviously, there's different kind of staging here. And this is, how many of you remember, I suppose some of you were born already into a world where mice have human ears on their backs, but some of you might remember seeing it for the first time. Yeah, so it was striking. It was, for me, it was a watershed event. This is when I said, okay, I want to understand this technology. I want to figure out what it means. What does it mean to engage? What does it mean to sculpt life in such a way? One of the science, so I, actually 3 years ago, I curated a show to commemorate the 20th anniversary of the appearance of the mouse feature Nuremberg, looking at the cultural impact, commissioning artists, looking at artists already done work in this field, and looking at some popular culture reference to that, and there's heaps of them, everything from The Simpsons to South Park to CSI to many, many other television shows that would constantly show this mouse with Ehrenbeck as a reference. But I also went and collected oral history from those scientists, really interested in seeing if they had any artistic kind of reference, if they wanted to somehow refer to the work of Hieronymus Bosch or Dalí or any one of those. And obviously, when it comes to medical people, they would tend to say, you know, we just want to do good in the world. We just want to fix people. Don't put words in our mouths. But when I probed them a bit further, they said, you know, we were trying to get funding from the Department of Defense. We knew that if we would put a knee on the back of the mouse, no one would say anything. So we decided to put something which is recognizably human and an extremely complex geometrical shape, an extremely complex sculptural form to impress our potential funders. So they knew the impact of that. However, 3 out of the 4 collaborators, when the BBC documentary crew came over, decided not to show it to them. And one of them, Charles Vacanti, decided to show it. And his story is actually quite interesting. He said after he finished the interview, he called the reporter and said, do you want to see something cool? And she said, yes. And he takes her to a back room and shows her the mouse. And then she says, oh, bring the cameras, we want to shoot it. And then they staged this image and did this whole thing about it. Now, Vacanti, jokingly or not, said, I didn't really thought it's going to make anything because it's, you know, it's the BBC. Who heard about the BBC? It's like, you know, no one would notice. And obviously, overnight, it became a sensation. And once you realize that actually he's holding more than just a scientific experiment, it was a cultural object. He cut the ear from the back of the mouse, and cast it in resin as an object, a museum-ready object. Those of you who do science, you know that there's no scientific data that you can get from something like that anymore. It became an object for contemplation, an object for a museum. He was trying to flog it to the Smithsonian. I'm going to the Smithsonian next week, and I'll ask them why they didn't want to take it. So it's still sitting in his home. Last time I inquired, he wants to sell it for between $2 and $4 million. So if you have some spare change, you can knock on his door and get this really important object. I think he did a massive mistake by not keeping the mouse there, which is kind of really strange. But the actual person who did it was a Chinese plastic surgeon called Yilin Chao. And Yilin Chao, when he went back to China as a hero, got $350 million from the Chinese government to set up the Institute for Tissue Engineering in Shanghai, and then he also was commissioned by the newly formed Museum for Science and Technology in Shanghai, and he created a mouse with ear on spec specifically for the museum. As far as I understand, in conversation with him and the curators over there, the mouse was alive in the museum for about 2 weeks, walking around with the mouse, with the ear on its back, and then by the end of that process, the mouse was plastinized. And actually, this image is quite interesting because I went there quite a few years back, and then I went there in 2015 as part of my research. It was under a dome, and it was full of dust and fingerprints, and I said, like, this is not really very respectful for the object. It's a really important cultural object. And they lift the lid, so this is the first time that the lid was lifted. I was able to take some photographs. They cleaned the dome and put it back there. But then, interesting enough, Vacanti decided that this is really a cultural object, so it's too important to leave others the control over, and he decided in 2006, so 11 years after the first appearance, he decided to do something that very, very few scientists do. He decided to copyright any image or any depiction in any media of an image of the mouse that has a human ear on its back, and here you see. So this is from the American Copyright Agency. You can see that he copyrighted a sculpture of a mouse with ear on its back, a photo, and a drawing. Yeah. And when I worked on the exhibition and I interviewed him, out of the blue he says, I give you a permission to use images of mice with ears. I said, like, what? It's like, it's not up to you. He says, I copyrighted that. And I consulted with lawyers. They said, there's no way these copyrights hold. But in his mind, and this is kind of an interesting thing, in his mind, he's the holder for at least 99 years after his death of our imagination of mice with ears, with human ears growing from their back. So, you know, something really strange is happening. Those objects are becoming really strange objects. The idea of strangeness is not now the realm of art. It's actually the realm of very many other disciplines. And there is an increasing awareness of those disciplines, of the strangeness of their doings. Dolly is a great example, but Dolly is not so strange because Dolly looks like any other farm animal. How you can make Dolly special? Dolly lives now, or she doesn't live, but she's stuffed and sits on a big rotating petri dish in the National Museum of Scotland in Edinburgh. And the interesting story there in my conversations with the curators there is that she's being looked after by the natural history curators, but she's positioned in the technology section next to the oldest computers and planes and rockets and kind of... So this kind of disconnect, and this is, I'm working at the moment on a really big project of what does it mean when we have objects that are crossing the cultural and natural history divide. But there's another thing about Dolly, because Dolly was such an important event in the history of technology that many other museums wanted a part of Dolly. Yeah, so I was lucky enough to go to the welcome wing at the Science Museum in London when they had a special exhibition where Dolly's droppings were presented there. Yeah? So if you really want to fetishize those technological approaches to life, you can go and worship Dolly's shit. Yeah? But what you can't do is ever encounter those 277 failed attempts that happened before Dolly was born. Yeah, so there's a very, history belongs to the winners. Yeah, so you can even have the winners' shit, but not the losers' bodies. So the Natural History Museum are really odd places. This is from, and I'm sorry, the quality of some of the images I'm going to show you are not that well because I've been taking photographs in museums that you're not allowed to take photographs in. So I had to sneak. This is from the Natural History Museum of Nantes, and you can see how the idea of the old-style Natural History Museum is to privilege the form of the organism, yeah, in most cases. When it comes to humans, you have this strange cabinet of oddities. But when it comes to the traditional idea of the Natural History Museum, you have an idealized form because the only thing that belongs to the original organism is the hide. And then it's being stretched on an armature of the idealized form of the organism in some kind of an action type of sculpture. Yeah? So it shows the animal is kind of in movement, but it's not, there's no animal inside. Now it's basically polystyrene. But at least, you know, you have some connection. You understand what you're experiencing as a visitor to the museum. However, the collection of museums moves more and more towards things like that, where museums are collecting DNA and tissue samples. And this is what I refer to as the fetishization of the information embedded in the potential of the DNA to become that organism. Yeah? And this is kind of a totally decontextualized idea because now we know things like the microbiome. They're not collecting the microbiome of the organism there. They're only collecting the DNA of kind of the prime example of that specimen, which makes very little sense and obviously makes very little enjoyment for the visitors. Yeah? So they don't really, it's really hard to connect with a bunch of vials in a freezer. So United and myself have been talking a lot about this idea that we're starting to see this new type of fetishization of life, yeah, through technological means. Yeah? So living fragments, but not just living fragments, those forms of lab-grown life require different epistemological and ontological understanding and, by extension, a different taxonomy. And this idea of taxonomy, where you classify those new life forms that are created, that are being fetishized. This lamininity of this kind of technology approach can lead to a form of fetishization, which we refer to as neo-lifism. So another example, I went to visit the Natural History Museum in Vienna, and again, there are images, you're not allowed to take photographs there. The Natural History Museum in Vienna is a really interesting place because it's the museum of a museum. It's a World Heritage Site. About 80% of the museum have to stay in the original form of the 17th century 1700s museum as it was formed. So the curators, they have to maintain the collection and the display as it used to be. And they only have about 20% of the museum space to have changing exhibitions. And this was one of them where it was an exhibition that was commemorating the 150th anniversary of the publication of The Origin of Species, and they were trying to find new ways to engage with it. You can see my friend Frankenstein over there, or actually his monster. You can see the babushka doll, but you can obviously see things like the death masks of Dolly, and you can see our friend the mouse with earrings back on the stage on that petri dish. But you also had a section about the future, and one of the sections in the future, and I don't know how many of you read German, I don't, but I can understand that they refer to transgenic art. Yeah? And there's a reference to Oron Catts that was mentioned earlier, and his work. Now, Oron Catts, some of you might know, created or claimed to create a green fluorescent bunny called Alba in 2000. The bunny was never been seen because it was never released from the lab, so it lives in our collective imagination. And obviously, the museum couldn't have access to that rabbit, so they decided to take one of the most scruffiest hairs from their collection and shine a green light on it, standing for Alba, yeah? And even in the catalog of that exhibition, they have that image rather than Eduardo's original one, because speaking to Eduardo, they never asked permission from him. I had dinner with the director of the museum and asked him, what the fuck? And he says, you know, now you artists are moving into our world, we can move into your world. So this is where the collapse of those contracts, where you can see that those kind of realignment of forces are taking place in a very, very strange way, and we need to try and make sense of it. Another example, which very briefly, I don't know how many of you have seen these nude chickens. They were developed in Israel as a way of creating chickens that can grow in hot climates, because those broiler chickens eat about 80% of the body weight every day. They generate a lot of heat, and in hot climates, they would just die of heat exhaustion. So this geneticist, who was a very traditional geneticist using traditional crossbreeding and selective breeding techniques, managed to breed this kind of chickens. Now, it was a public relations disaster. I won't go into the story, but the story came out on page 3 of the biggest Israeli newspaper, where they usually get Israeli soldiers in bikinis, and they decided this time to show a naked chicken. And obviously, the media was all over it. The companies that signed the contract with this geneticist pulled out because they said we can't deal with this disaster. So now those chickens are being hired out for artistic purposes. So something is strange. Yeah? You should admit that it's messed up. So we decided to set up a lab to engage with it. So Symbiotica is this research center at the University of Western Australia where we get artists to engage in the most experiential way with the tools of modern biology as a way to engage with those questions around life. So we see ourselves as a place that it's dedicated to the research, learning, critique, and most importantly, this hands-on engagement with the life sciences. Yeah, we're trying to find a new language. We're trying to articulate those freaks, yeah, how we kind of make sense of them. So what we do, we explore those philosophical ideas around life. We identify, so we basically troll through scientific literature and try to find areas that need cultural scrutiny, and we do so by basically trying to replicate those experiments, try to recontextualize them within artistic context. We also try to develop, so in a sense, I suppose you agree with me by now that life becomes a raw material, biology is engineering, so we do have, for better or for worse, a new palette for artistic manipulation, where life is both the subject and the object of our manipulation. Yeah? So we are looking at it also from a very kind of formalistic and material perspective. What does it mean to engage with life as a material in such a way? And we do develop our own technologies, and I'll show you some examples as well. So sometimes the artistic needs are very, very different from the scientific needs, so we design our own bioreactors, we design our own environment for research and display of art, of living biological materials within a cultural context. And when it comes to within a cultural context, one of the hardest things that we need to deal with is actually introducing those living biological artworks within an institution like this one. Museums are set to be places of death. Yeah? They're places that are designed to keep dead things as dead as possible for as long as possible. And then you bring life into it. And the amounts of phone calls that I would receive from conservators of the museum, they would say, hey, you know, your work looks differently today than it looked a couple of days ago. And say, like, yeah, of course, it's alive, it's changing. It's like, no, but we can't, we've signed, you know, the condition report, and we promised you nothing would change. I said, no, you can't. It's like, this is life. So anyway, we run academic programs, we run exhibitions, and a residency program. We run a big triennial, so we just finished our second triennial, which is called Unhollowed Arts, and we published a book. You can find it on Amazon if you want to pay money. But we really try to kind of engage, and this triennial was dedicated to the bicentenary of the publication of Frankenstein. I have really beautiful stories about Frankenstein I won't tell you today, but if you're interested, you can find them in the book. And in a sense, what we're trying to do, we're trying to engage with life in all of its manifestations. So one of the things we say is that we are, and I talked a lot about it in the last couple of days, about the poverty of our language. The fact is that we only have one word to describe life in all of its manifestation. We can't, it's such a blunt instrument. We can't engage with the nuances of the different levels of life, of the engagement with life, of what we learn about life. We're really using a very blunt instrument to try and engage with it. And just to contrast it, in the English language, we have 50 words to describe excrements, yeah, in different manifestations. So we can be very nuanced about what's coming out of our bodies, but we can't deal with what our bodies are, the essence, in a sense, which is life. So this is exactly what we're trying to engage. We engage with life from the submolecular to the ecological. We have a biological lab, which is ours, so it's a level 2 biological lab, so people who come to do research with us are need to commit to spend time in the lab and engage in an experiential way. We train them, so we don't really talk much about our work in the context of art and science collaboration. It's more about mentorship, so the artists are being mentored by scientists to develop the skills they need to develop the work themselves. And when it's come to life, it's really important because it's about the responsibility that one needs to exercise where they're starting to engage with that. So it's not about commissioning someone else to do the work on their behalf. As I said, we run an academic program. One of the things we run is what we refer to as the real MBA, the useful one, the Masters of Biological Arts, and it's open for business. But now let me talk a little bit about my own practice. And I'm aware of the fact that I only reached slide 37 out of 142 that I have here. So Boris, you tell me when I need to stop. Yeah. So my background is actually product design. So I studied product design in the early 90s, trying to imagine a future. So I was doing then what is now known as speculative design. No one used this term at the time. So I was speculating about a future where designers would be called upon to design living biotechnological products. And I used, obviously, computer-generated imagery, and I was trying to figure out, because I never really spent time in the lab. But then this mouse with the ear on spec hit the media, and I said, okay, tissue engineering, this technology where that created the ear on the back of the mouse is really what I want to concentrate on, because it's so rich. And I felt that there's no point of me to continue my research as a designer, because I was way more interested in the questions that needs to be addressed before we can start to think about those things as products. So tissue engineering is a really interesting, and the whole field of regenerative medicine, and we have few regenerative medicine people here, is an extremely interesting thing because it really changes the body and our perception of bodies and ideas about the bodies. I was fortunate enough to spend a year in Harvard Medical School with Joseph Vacanti, one of the scientists who was responsible for the mouse return spec, the brother that didn't want to show the ear. And he invited me to spend, and Yonat, to spend a year with him. And this slide is actually from a presentation that he gave for the 3rd International Tissue Engineering meeting in the year 2000, appropriately in Disney World in Orlando. So we were served breakfast by Mickey Mouse, and then in the afternoon we were talking about human mice on ears, and I'm sure it was all planned. But what he was showing is this amazing conceptual shift. So this image over there is how we imagine we're going to repair the body in the 1980s. 10 years later, everything changed. Yeah? So if in the 80s we're talking about replacing ailing or missing body parts with hard and dry technology, things are changing. Suddenly the body becomes its own technology. The dream of regenerative biology and medicine is to somehow harness those latent regenerative powers of the body and make the body into its own technology. The original idea of tissue engineering, the way the ear on the back of the mouse was done, was very straightforward, and actually every artist can understand how it works. It's like a loss wax system where you start with a degradable polymer in the desired shape of the organ you're trying to replace. You seed it with the appropriate cells. This is, again, a slide from Vacanti's presentation. You grow it in vitro outside of the body for a while, and then you implant it into the body, and here you have a new body part. Now, in 1995, there was an international consortium of scientists that claimed that in 10 years' time they'll be able to grow a fully functioning heart. Yeah, we're not even close to achieving that. We're way past the deadline. But the mindset that we might be able to grow a fully functioning heart that will wait for us on the shelves in the hospital is still what drives a lot of this research. And for me, this mindset also generated kind of a reverse logic. So if in the 80s we were told that we can replace the heart with a pump, and in the 90s we were told we can grow hearts, my question was, why not just grow pumps? Yeah? Forget about reintroducing this functional tissue into bodies. Why not create what I later started to call semi-living products? So products which are made out of living tissue. I was extremely naive then, I must admit. I was thinking I'm going to save the world. I was thinking that by using biological processes, not just mimicking biological processes, but actually using biological processes, make them more compatible with the living world around us. We can generate a society where there will be no waste, where we'll care for our products as if we care for our plants in our garden, and that we would be able to generate something which is much more sustainable and compatible with the world we live in. It's an extremely naive view, I must admit, and I don't think that anymore. But many companies, many startup companies are now selling this narrative, as if this is what's going to save us. So in 1996, Yonat Zur and myself joined forces and developed the Tissue Culture and Art Project, the very first institution we ended up working with. So we knocked on the door of a scientist, and to our surprise, they invited us and said, come and work with us. It was called the Lion's Eye Institute in Perth. It was an eye research institute. And we entered the lab. And okay, warning, the next image, the next video might be a bit disturbing. This is what we are confronted by. So every afternoon, or not every afternoon, twice a week in the afternoon, we would get those boxes, those cardboard boxes with those half rabbits' heads. We were told the rabbits were killed for food in the morning. The heads were then sent to a brain research institute. They would cut the heads in half, take the brains out. And the research lab where we worked would get those half rabbits' heads. We would then take the eyes out from the rabbits and then put them in this vial with antibiotic solution overnight. Go and have a chat with the scientists we're working with, and he was like, I'm just a graphic designer, I'm just a genetic designer, leave me alone. But then the morning after, we would look down the microscope and start culturing the cells, and we would have living cells. And that's when we started to realize that also our language is lacking, because obviously we were starting with something that, for all intended purposes, was a piece of dead meat. Yeah? You can't get more dead than being a half-rabbit head with no brain. Yeah? But 24 hours later, we had living cells that we could culture, we could grow, and we would start to use them for our art. So we started to grow those skin cells, they were epithelial cells, over those glass figurines that represented different aspects of human technology. This one is an interesting one. We started with something that we thought would look like a bomb and ended up looking more like a fertility goddess. And this is kind of a strange occurrence. But we were not satisfied with that. That's not really what we were interested in. And then in 2000, we were lucky to be invited to present our work in one of the biggest art and technology festivals in the world, Ars Electronica in Linz in Austria. And they were brave enough or stupid enough to, and they had enough money to invite us and challenge us to show living tissue-engineered sculptures in a cultural context. So we decided to grow our own version of the Guatemalan worry dolls. Those are small dolls you give to kids before they go to bed. The kids tell the worries to the dolls, and the doll's supposed to take the worries away. So we thought it's kind of appropriate that the first tissue-engineered sculptures would engage also with the anxiety that we feel around kind of this strangeness of life. So this is how the installation looked like. It was a really interesting lesson for us because that was a case where the technological frame took over. I can guarantee that about 90% of the people who saw that installation saw a bunch of artists taking scientific equipment and putting it in a cultural context, and that's all. So they couldn't see the worry dolls, which were admittedly very small, but those are the worry dolls, kind of the before and after images as they were growing. We would grow them in those microgravity bioreactors, and we also started to deal with what we started to refer to as the aesthetics of care, where we would come every day to feed our sculptures, almost like the feeding time at the zoo. We also had a guest book where people could write worries to the dolls. We hoped that people would express their anxieties in regard to kind of the future of biotechnology, only to find out that many people expressed the most intimate anxieties and worries, and most kind of, to such an extent that when we posted it online, we got quite a lot of people coming back to us and saying, no, it was between us and the dolls, we don't want it to be public, which is fair enough. In later stages, so we showed this work for quite a few different times. We started to design different labs, different environments. We introduced a microphone where people can actually whisper to the dolls. This lab from the Science Gallery in Dublin in 2011, we designed like the House of Dorothy from The Wizard of Oz. So it was like tilted. The only thing was missing was the ruby shoe. But it was kind of the sense of taking something out of context and putting it there, but also starting to create those portholes, those frames where people can start to see the specific artworks. Okay. We also introduced this idea of the killing ritual. So we started to also talk about the fact the responsibility of artists who are working with manipulated living matter is also to be very transparent and talk about what happens to those life forms in the end of the show when we are not there to look after them. So we would create those public executions, if you like, where we would invite the audience to come and kill the sculptures by touching it. And many people told us that by touching the sculptures, that's when they started to believe us that we actually done what we claim we were doing. I have to skip quite a few projects. This is a recent project or collaboration with the Center for Synthetic Mammalian Biology in Edinburgh, where we're trying to fuse a human and a yeast cell to create a hybrid organism that live across kingdoms and doesn't have any classification. We're working with things like hybridomas. Those are cell lines that you can mail order from the American Tissue Culture Collection, where they are basically made out of 2 or more different organisms. Here you see this is a mouse, a hamster, and another mouse. So those are 3 organisms created in one cell line that exists as a new organism that lives in the lab. This is a human mouse cell line for the creation of antibodies. And we then show them along kind of this idea of the return of the cabinet of curiosity, starting with a 2-headed bird and then showing a traditional natural history display. And then the only living part of the display includes those hybridoma cells within this technological frame, so totally abstracted. So how much time do I have? We have time to talk about my... Fine. Okay, so I'll talk to you about the meat project because I think that's an interesting one. This is a project that might end up here in town in some stage in the next few years, so I won't talk about it now. I won't talk about Leduc and the mechanism of life, but that's kind of interesting also that synthetic biology has been around for more than 100 years. I won't talk about our compost incubators, that we're now creating those incubators where some life forms look after other life forms. And I'll talk about meat just for a second. So in 2000, Jonathan and myself, when we were residents in Harvard, we realized that we should really engage with the idea of meat. If we deal with life, if we deal with the relationship we had with other life forms, there's no more intimate relationship with another life form than consuming it and making it part of your own body. So we kind of speculated, what does it mean to be able to eat meat that was never a body? Yeah? What kind of relationship? If you are what you eat, what is that going to make us if we're starting to eat meat with no bodies? Okay, I'll skip the video. This is how it's being made. In 2000, we were not able to eat the very first piece of meat because it was done within a biomedical research lab. We didn't have a license to grow meat. In 2003, we were invited to the show in France, and they had enough money to keep us there for 3 months. We were able to get brand new equipment, so there was no chances of cross-contamination. And we played on the idea, super gourmet. Oh, sorry. We played on the idea of what constitutes foul food. We knew that French people don't like the idea of eating engineered food very much, and most other culture.
David
Oh.
Mullen, yeah.
Doesn't work. Okay. We knew that many other.
And we'll start by killing the steak and eating them. And it's an extreme never cuisine.
I don't know why it's doing it. Okay, never mind. So anyway, the video actually, as far as we know, it's a very historical image because, as far as we know, this is the first time that anyone was putting a piece of meat in the mouth that never came from an animal. So just to finish the story, we were using frog cells because we knew that French people like frog meat but don't like engineered meat. So we combined those two foul bits of food and generated tissue-engineered frog meat. We grew it for about 3 months. The problem, we then cooked it in this nouveau cuisine dinner. We were able to grow about between 5 and 10 grams. We shared it among 6 people in the audience, and actually the curator and a few others. And the sauce was really good. I was a bit concerned about health and safety issues, so I asked the chef to cook it in a garlic and honey sauce, just well-known antibacterial agents. But the texture was horrendous. The polymer that we were using didn't break out completely because we were confined by the duration of the show. And the frog cells, they were muscle cells, but we didn't exercise them, so they had a bit of a jelly-like texture. So you can imagine trying to suck on and eat jelly on fabric. And to our delight, 3 of the 6 participants in the dinner, I swallowed it because I put my mouth where my money is. But 3 of the other participants couldn't swallow it, spat it out, which was great because we could then collect them and put them in the follow-up exhibition, The Remains of Disembodied Cuisine, where we had the video installation following the whole process plus the bits that were spat out. So I have so many other stories to tell you, but I suppose maybe I should stop now, or you want to hear about leather? One more story, okay. I'll skip that. I do host cooking shows now where I invite scientists and philosophers to cook against each other, to talk about the future of life. And actually, recently we also started to get some nice chefs. So I had a recent one in Dublin where I had one of the finalists of MasterChef Ireland cooking for us as well, but using her breast milk. She just had a baby. So, but our secret ingredient is fetal calf serum. Now, fetal calf serum is what you need to use when you grow many of the cells, and especially when you grow fast metabolizing cells like mast cells. When you want to get large quantities of them, you usually use between 10 and 20% of the nutrients of the food for those cells, which comes from the blood plasma of unborn cows. So anyone who tells you that they managed to grow victimless meat in the lab is telling you stories up to this stage. There's about scientists who are trying for the last 60-odd years to find a replacement. They really hate this substance, not just for ethical reasons, but because every batch is different. And they still need to use it because it's got about 40,000 different components. Scientists were able to isolate about 20,000 of them, or 30,000 of them maybe, and we're able to synthesize about half of it. But there's still quite a lot of unknown substances there that still seems to be needed for cell growth. So all of those people who tell you that they're growing meat in the lab, even those extreme vegans, they still use this. Yeah? So watch this space. Maybe they'll come up with a solution, but not anytime soon. All right, that's the first cooking show that I did with Mark Post, who is the one who claimed to be the first to grow the burger. So that's in 2013. This show was exactly a year, so I trained him in kind of public presentations. This is kind of his show, so this is kind of a nice example of science imitates art. Yeah, so he did a food performance, again, really hard to verify what he did there, but that was done with a much higher production value than what we can do, but that's cool. We then started to grow insect meat, so we created our stir fly, so we had a fly in our soup, but it was contaminated by fungi, so it became a mushroom soup and couldn't really eat it. So the leather project is kind of an interesting one. It's United and myself, we grown our first piece of leather in 2004. We actually, as opposed to many of the other experiments we're doing, that was, we had a bunch of honor students from the anatomy school, and they were also testing if our hypothesis was right, and we were able to grow a leather-like material. This piece is already 14 years old, and it's still in our lab and still very much like leather. We managed to, how do you call it? Fix it, and and stain it, and it does look like leather. But when we showed the live version, we decided to go and kind of engage in this idea of having a tiny jacket that can fit a mouse growing inside this artificial body, this techno-scientific body. It was really interested kind of reversing the roles. So rather than the leather being a protective cover, this leather was protected from the world by the technology, and that allowed it to grow. In 2008, we were invited to show it as a part of the Design Elastic Mind show at the MoMA in New York, and halfway through the show, we died. There were about 250 different works in the show. It was supposed to be a fairly positive show about this great future. If we get artists and designers to work with scientists and technologists, we're going to fix all of the problems of the world. And the only living thing in the solid installation, in the exhibition, died halfway through the show. Paola Antonelli, the curator of MoMA, was actually very generous in her kind of description of the process and the kind of the problems associated with it, and we felt that it's actually generated a way more interesting conversation. Obviously, the media had a field day, so murder in MoMA, MoMA kills art. But in a sense, this is kind of where, so what's quite interesting in this context, it was considered to be a design failure, but as an artist, it was one of the best interventions that we had. And as far as we know, this was the second time that MoMA was showing something living after Steichen in 1936. So thank you very much.
Now,
I still have heaps of time.
I suggest we have one question, and then we invite the panel to the stage. Is there a question?
Over there in the back.
Microphone there.
How do you feed the muscle cells to grow? Like, what do you feed the muscles?
So we feed them with, basically, so this is another really interesting thing, and again, in a conversation around growing meat in the lab, is kind of, it's a really strange one, because you basically pre-digest the food for them. So it's similar to the nutrients that are running in your blood. So when you think about the amazing way of growing meat in the lab as an environmental solution, you have to question it, because you need to outsource the digestion to another thing. And in a sense, what you need to provide the cells is all of the sugars, all of the amino acids, all of the other nutrients, all of the vitamins, all of the minerals, all of the hormones, anything that the cells need, all of the signals they get, all of the nutrients that they get in the body when they grow, yeah? Which includes also between 10 and 20% of the fetal calf serum at the stage of expanding those cells. Because what you do, you grow cells which are called satellite cells, and those are the progenitor cells for the muscle cells, because once they mature to muscles, what's referred to as terminally differentiated, they just stop dividing. So you need to expand them really, really fast in really big quantities. And it is an exponential growth, so they can grow quite fast. If you have enough nutrients and enough incubators, enough space, yeah, you start with 2 cells, 4, 8, you can grow the whole world in a fairly relatively, and this is kind of the rhetoric that they're using about how efficient it is. But you have to remember that you also have to feed them with what would allow them to grow, and you have to clean the shit, which is something they rarely talk about, the waste that is generated. So you need all of the functions of the body have to be outsourced to technology in order to grow muscles in the lab. Yeah. And that's one of the critiques I have of this promise of salvation through cellular agriculture, this idea that the lab is this magical place where you put stem cells on one hand and you feed the world on the other.
Can I ask an addendum question to that, which would be about the muscles? How do you exercise them?
This is a really great question. Let's see if I can jump there. So yeah, you need to use there's different, Mark Post was using, a system which was basically putting tiny Velcro on the edges of the muscle fiber and then exercising them. We were working with piezoelectric, so we were basically, this is a project which is called futile labor, where we were growing muscle cells as an exercise in futile labor, if you like. And we were trying to transmit also this experience. A lot of our work as artists is to try to create other forms of mediation. We don't like microscopes very much because we want to get the audience. So in this case, the vibrations of the cells as they were stretched was translated to subwoofers, so people could actually feel them in the body as they were approaching this installation, and they would get live kind of vibrations from the muscles manifested in the bodies as subsonic. Yeah. Make sense? Yeah, there's many other ways to do it, but it's still very expensive, very complicated, and not very efficient. Yeah.
panel is invited.
Let me just... So, oops, sorry. I just want to, we'll have that as well. Food for thought.
Okay, I'll present our panel. Brenda Ogley is a professor in the Department of Biomedical Engineering and the head of the System Regeneration Lab that investigates mechanisms of stem cell differentiation in the context of the cardiovascular system. I'm not used to pronounce all those words, as you can see, and develops new technologies that advance stem cell research and promote its translation into the clinical practice. Mark Barella is a historian of biology with interests in evolutionary theory, genetics, behavior, and the environment, who studies interpretation and application of evolutionary theory from the late 19th century to the present. Currently, Mark examines the connection of group selection to ethology and evolutionary psychology. Steve Kelly is a senior fellow at the Humphrey School of Public Affairs. Prior to joining the U, Steve served as a public member of the Minnesota Board of Medical Practice, in the Minnesota House of Representatives, and in the Minnesota Senate, where, among other things, he has introduced a bill promoting stem cell research. And I planned to kind of see the conversation in a completely different way, but now I'm thinking about something else. You started the lecture with babies on the freak show, and you said that back at the time, there was just no other cultural solution for this. You just culturally didn't have any other place to place them. And I wonder whether we are in a similar situation now, sitting in the Art Museum, because culturally there is no other place where we can have this conversation. And we need to have people with 5 different titles on the stage, because culturally we cannot have this conversation within one discourse. We actually should bring 5 different discourses to the stage to understand what's going on.
So thank you, Boris, for the invitation. Do you want us to turn it on? Okay. Thank you for the invitation, Boris, and thank you, Oron, for a stimulating talk. And more than an answer to your question, Boris, it's I guess a question back to Oron. So from a public policy, political perspective, when you started with mindsets and institutions, those are things that public policy works on. And so I'm curious how you see things progressing, because I agree that to a certain extent we lack the cultural space, we lack the language to talk about it. Do you feel like you've made progress? And if so, what else do we need to make more progress in changing mindsets or creating the cultural resources that we need in order to deal with the kinds of questions you're raising?
Yeah, this is an extremely interesting question, and it is kind of something that I'm grappling with as well, because first of all, starting this project, I never imagined the impact that it's going to have. So our work has been submitted to American Congress as a case study. Laurie Andrews, the lawyer from Chicago, who was part of the advisory committee to bioethics to Clinton, was using our work as an example for that. Our work was submitted to the EU for consideration as well, as a sign for things that needs to be dealt with. Actually, even Michael Crichton wrote about our work in his book Next, which was part of that. So Margaret Atwood is referring to our work as part of her Mad Adam conversation. So it is reached places that I would never imagine my work would ever reach, especially based in kind of the most isolated capital city in the world. But here we are doing that. But saying that also, last year I was in New York. There's a company that is trying to grow leather and meat in the lab. They set up a pop-up shop in Soho where they were trying to sell the idea of lab-grown leather. interesting enough that they moved from animal culture to genetically engineered yeast that produces collagen, but they did this timeline. That's how they reached this point, and our work was part of this timeline. Now, I don't know if I should take it as a compliment or as an insult, because this wasn't what we were trying to do. Our work, in a sense, and we talked about it with Michael the other day, this idea that artists are gentrifiers, even when they're trying to do social good. Yeah? So you move them into a bad neighborhood to help the local people, they increase the value of the property, and they mess up. Yeah? So this is something that always, in a sense, happens. So art can always be used even against the intention of the artist. But because my work in very early stages, I realized that my work deals with life mostly, but also with questions around control. And I decided that there's no way I can take control over my narrative and my art once I released it out to the public domain. So if the only thing I can do as an artist is to make people aware of things and make them think about it. I'm not positioned where I can tell them what they ought to think about it, but I want to put them in a position where their perceptions are being challenged. The other thing that I find actually being really successful in the sense that I can go in and contaminate scientists' minds. So I found myself giving keynotes in some of the biggest scientific conferences. I was asked to give a plenary in the SB7 meeting, so it's one of the biggest synthetic biology meetings in the world in Singapore, and that allowed me to at least show them that there's other ways to think about the very stuff they're doing. I'm talking about pointing the finger at those ontological breaches that they're creating without even understanding that they're doing so. And that's all I can do.
Because we have had a chance to visit over the last couple of days, and one of the things that I noted was that this art has the capacity to shock, to provoke strong visceral reactions in all of us, and then from a public policy perspective, I would love to see us move beyond that shock and consider the role of art or the artist in helping us to process past the shock into a more nuanced approach to the questions we have to answer.
So I just want to comment about that. So you're totally right, actually, that the name of our retrospective show in 2011 was Visceral because it was all about that. But the visceral reaction is exactly the reaction that indicates that there's something that needs to be addressed. Yeah, so we need to go. This is exactly the area that I refer to, the area where we have no words to describe, so we feel physically kind of sometimes even sick when we encounter something that is almost in contradiction of our long-held beliefs. Yeah. So as you say, we need to go over it. Now, the approach that we had is that we are doing it ourselves. So we can't be righteous about it. We are part of the biotechnological projects. We are working with stem cells. We do IPS. We do those kind of things. We engage with it in the most experiential way. So there's no way we can wash our hands and say we are pure in some way. We're actually part of that conversation in the most intimate way. So, in a sense, as I often tell the scientists, I'm more than willing to be the freak in the show, so you will be shielded, so we can start to have a more nuanced conversation later on.
I think just to add on that from a scientist perspective, I think that this is an important time of conversation, of really thinking about the issues beyond the issues of what can we do we want to do it, and why, what direction are we headed. And I think one of the things that I see, I haven't really thought about it in this context, so I'm really appreciative of that perspective. And I think building on that is the information divide and thinking about how that is conquered. I think right now, bioethicists have come on board and are on the scene, and I think that's great, and we discuss things in the context of a scientific community. But I think the conversation could be much richer in the context of the community, of what it means for us as a society. And I think that's what, at least from my perspective, I'm hoping that a lot of what your work is doing does, and I think it is.
Did you want to say something?
No.
Okay. So as a historian, I was really interested to see, particularly, and we talked a little bit about at lunch yesterday with Jacques Loeb's work at the beginning of the 20th century and the end of the 19th century, and this concept of engineering and control and how early that develops for these biologists and how they have this image. And I wondered if you wanted to say maybe a little bit more about what concerns you have about control, because I think for scientists, there are these ethical kinds of concerns. There are these concerns about, well, do no harm, As artists, maybe you have a bit more sort of free play to say, well, we're not trying to cure a disease, we're trying to provoke conversation or demonstrate a technique or something. So I just wondered if you'd say a bit more about that whole.
I had to skip really fast on the piece that we've done around Stéphane Leduc. So Stéphane Leduc was a French scientist in the turn of the 20th century. He was fighting the vitalists. So there was a deep philosophical conversation between the materialists, those that believe that life is just another kind of material manifestation of natural processes or material processes, and the vitalists who that basically believe that there is some kind of special life force that cannot be reduced to material. And as a way for him to prove it, he wrote a book called The Mechanism of Life. And in chapter 10 of that book, he coined the term synthetic biology. And he says then, 1911, we know enough about life that we can start to synthesize it. Yeah. And the problem there is obviously in retrospect, 100 years later, we say, like, you knew nothing then. Yeah. But you had the hybris of thinking that you can control life and the mindset. And this is another thing that we need to remember. Some of the conversations we have now have been manifested in some different ways in the early 20th century when people started to feel that they can control life. Yeah, and we know what the manifestations of that were. And there was this big reboot moment of the Second World War where we wiped the history. So I'm sure that 90% of you never heard about Alexis Carrel, even though he was the Nobel Prize winner. When you go to the Rockefeller Institute and they have the flags with all of the Nobel Prize winners from the institute, Carrel is not there. He was wiped out of history because we didn't want to learn the lesson. Yeah? So in a sense, there's this cultural amnesia and this idea that you know, one of the most amazing things about the Human Genome Project is promise that once we have the human genome, we'll be able to have, we have the instruction book for a human, was the realization that that's not the case, that life is way more complicated than that. When Craig Venter created what he referred to as the minimal life form, he created a bacteria, which is funny enough was a derivative from, which was called the Mycoplasma genitalia, which is a parasitic bacteria that lives in the genitals of male primates. So there's some kind of appropriate story there, which I won't get into. But he then did a Microplasm Laboratorium, which was kind of a basic life form that had 372. Actually, no, Laboratorium was the one that he sequenced, but he created something based on that, which had 372 genes. He didn't know the function of at least a third of those genes. Yeah? So there's something there that we still don't understand about life, but the public this kind of perception of synthetic biology is that we know enough, similar to what Carrel and Laub were talking about, that we know enough and we can control it, you know. Right. And when Carrel was talking about creating a race of superhumans to control us and throw the dysfunctional people in gas chambers, that was a direct result of this mindset. You know, so some of the conversation we hear coming from especially Silicon Valley and California, the transhumanist rhetoric about, yeah, you know, this technology would enable us to reach this stage, this is rehearsing stories that already happened in the 1930s. And what we need to do, and what was interesting, and this is kind of where I think hopefully things are different, I couldn't find evidence of artists actually responding to that at the time. Yeah? So maybe the fact that artists are now doing this kind of work might change some of the discourse in some way or another. Right. So, yeah.
Okay, if there are no questions, I'll ask the question I did plan to ask. And then the clash of cultural contracts. And that has to do again with this merge of what we call disciplines in one place. And each one of those disciplines has different cultural contracts and different obligations. For example, you as an artist can do stuff for which Brenda could be kicked out of her profession. But once you create a lab when you both work together, how does that work and how that can work? What need to change to allow for such collaboration to happen?
So this is kind of an interesting thing. I have a whole different talk where I talk about those cultural contracts and the collapse of those contracts and the way artists are being coerced now to basically lose their role as the court gestures and actors promoters for questionable ideologies, but that's a different story. But what was really interesting with the setting up of Symbiotica is the rule that we decided to ourselves, and that we are only going to show what we are doing. So which resulted in what we refer to in many cases as the aesthetics of disappointment. And we're more than willing to engage. It's really hard for us to create spectacular works because the technology is not there yet, or might not ever be there. So we can't, when we've done our pig wings, we're able to grow, you know, tiny objects. We would never get a pig to fly. And this is kind of an interesting, we have a lot of conversation with the scientists, because some of the scientists says, you know, you really have to tell the story in a way that would capture the public imagination about the great work we're doing. And we say, your work is really great, but it's not that great in the sense, and there is this idea of honesty that is important for us. But let me put a caveat there, because as an artist, I never signed a contract with any of you that I'm going to tell you the truth. Yeah? So you would have to make your choice if you're going to believe what I'm saying or not. But I can tell you, don't believe me, that we decided that in Biotica, it's only the work that comes out of the lab that we can present rather than have those fantasies that many of the other artists in our field are engaged with, and they've got a full license to do so. Yeah? So the conversation when we have with the scientists is, first of all, it's like, you make us look bad in a sense because it's not as spectacular as we want you as an artist to be. Or sometimes you generate those conversations that we might not want to have. But in most times, it's like you are spot on. You are helping us to create a different idea about how science can operate in society, rather than have those hyperbole promises. It's about creating this conversation, which is the most important conversation about our relationship to this idea of life, about how far we are in now, how far can we go, and you're there at the edge of it, actually showing how disturbing it might be, how challenging it can get, but also how disappointing it might be compared to the rhetoric of the PR departments in our university. Yeah. Well, that triggered people.
There we go.
So actually, I
think they're recording it.
Please do wait for the microphone. Thank you. So I actually really appreciate what you said there, and it was something that I wasn't going to bring up, but you kind of opened the door. And you know, all of the examples are fairly precious items, you know, to scale. And the question that I have is, so what is the technical or budgetary limitation that doesn't allow, instead of having a jacket that's this size, a full jacket that is living, breathing, existing? What are the actual impediments to the scale?
So one of the main issue, and that's why you haven't seen hearts on shelves in hospital, for example, or kidneys or any other complex, you know, the only successful tissue engineered products that are there and that have been clinically proven are things like skin, cartridge, and a bladder, which is basically like a balloon. And that's the issue of thickness, because we still, scientists, you know, humanity, whatever, didn't manage to find the way to create a capillary system that is actually functional, that can be transported from the lab to the body in a way that would actually allow it to function. So the plumbing is the main issue to start with. The other issue is obviously, budgetary issues, but also the fact, that life kind of lends itself. Those cells, they don't like to be outside of the body. You know, it's not really their place. In order to trick them, you also change them to such an extent that when you reintroduce them into the body, sometimes the body even, and that's, I would love to hear you about it, because I was speaking to immunologists, so when it comes to therapeutics, sometimes even cells that are coming from the patient's own body would be rejected by the body because this is what our bodies do really well. You know, we all have pro-cancer in our bodies, and our immune system goes and cleans most of them out. So when the mouse with Ironsbeck was a great example as well. As far as I know, and this is still, it wasn't published, so 23 years ago, that image hit the media, the story was, and actually Charles Vacanti in that BBC documentary says, we've got the technology right, it's only regulatory issues that we need to deal with. There's still, as far as I know, Yilin Chow claims that there's one person that he finally managed to successfully grow an ear in such a way. The problem is that once you reintroduce it into the body goes and rearranges the cartridge because something is wrong, so you end up with this kind of shapeless blob rather than the shape of the ear. Joseph Vacanti was able to create a frame. Actually, I have another talk where I talk about the fact that he was able to create a titanium frame to hold it in place. But this is almost like an admission of the failure of the promise of regenerative medicine, because you need to put some kind of crutches to hold it in place. Yeah? So bodies are complex. Bodies don't work like that. This idea of being able to grow like a football kind of size skin, of the right thickness that would actually act as an insulator and maintain sterility on the inside. You know, there's so many issues there that are still very, very hard to solve. But, you know, I'm sure that if people put their mind to it and enough resources, it might happen, but then why? Yeah.
Why not?
Okay, that's an American attitude, yeah.
Well, I can maybe add to that just a little bit in terms of, so every tissue organ is different in terms of its criteria for success. And, part of cartilage or bone or skin is that it is constantly remodeled in the body. And so that's part of the failure. It's sort of interesting. We work in the cardiovascular space, and we're working with a type of stem cell-induced pluripotent stem cells that you alluded to. And then if you differentiate or derive mature cell types from those cells, we derive cardiomyocytes or cardiac muscle. One of the first sort of examples of that in transplant back into the donor of those iPS cells did see rejection, but then subsequent studies showed that it was part of some of the vectors that were used to introduce the genes initially. So now that hurdle has been overcome, and so there is still the promise of, or the potential for an autologous transplantation from iPS cells into a recipient or back into the person who was the original donor. But that said, there are certainly many challenges, and I think sort of coming back to where does the hype come from and where does the no hype come from? I mean, from a scientist's perspective, I think most of us are reining in all of the claims and trying to say, this is what we can do now. And I think, as you mentioned, in the hands of the PR and media, and maybe artists sometimes, it gets blown into a different realm that is hard to control. And I think that's part of the information that I was talking about earlier, is knowing what's possible now, what is the promise of the future or not, how far can we take it at this moment in time, and should we? So.
I should add that there's another, and actually I have a slide where I talk about the Grunter hype cycle. So this idea that every technological development goes through this hype cycle that we need to kind of come to terms with. It reaches the peak of inflated expectations, and then it goes down the kind of slope of disillusionment, and then some technologies pick up and some goes down the death valley. But the interesting thing when it comes to life and the promises of life and control, I think this is getting dangerous. So in a couple of weeks, I'll be in New York talking in a conference which is startup companies meet venture capitalists, talking about this kind of whole new era of which I refer to as biofabrication, the creation of consumer products, biotechnological-based consumer products. And what I find fascinating with those meetings is it's kind of, and this is part of the startup culture, of those ridiculous promises with those extremely seductive narratives in order to attract investors. And the investors know, that there's only one unicorn in the room, but they would throw money everywhere. The problem is that we, as the consuming public, because we're not consuming those products yet, we're consuming those narratives, we are collateral of that story. And we are collateral in the sense that we think that those stories are actually true, while actually the intention there is it's not really even directed to us. So somehow we have to be much more savvy about it. Somehow we need to figure out a culture where science is respected for what it does, not for what it promises, and the importance of science in explaining the world to us, but not expect too much and still support it. So I don't know how, and I think American culture is one of those. It's the most advanced in terms of science, but it's also the most problematic in terms of rhetoric and those kind of promissory type of imaginations and fantasies that somehow need to be reined in as a way to live in a world that actually is supporting the understanding of the world, but also not having those kind of creation of those fantastical narratives of control, because people and politicians like the idea of control. And even if you give them a fantasy, I talk a lot about this. There's a term coming from the psychology of gambling, which is called the illusion of control, where gamblers would throw dice and would do some kind of a ritual, thinking that it would control the results. And this is kind of where we are now. Yeah? So when our politicians have those illusions, someone tells them that actually soon enough you'll be able to control, you can have biometrics information, you can have all of those information. There is the perspective of creating, there's stories about China becoming a digital dictatorship. What a biotechnological dictatorship is going to look like.
Yeah, I have changing maybe a little bit, but I recall, I guess it was about 10 years ago now, when I was involved in discussions with Oron Catts, who did a project here, and with faculty members who were doing research in plant genomics, I think. And it was interesting to me because it's interesting that you talk about the artist being more free. And it became very clear through those discussions with faculty that that what they did not want was the artists showing what they were doing, and that it was okay if they did it for the sake of science, but it was not okay to do the same thing for the sake of art. So there was definitely a hierarchy there. And maybe that's old thinking, but I'm wondering if anybody would like to comment on that idea, if it still exists, or if you found that it doesn't exist anymore.
I can answer, but if anyone of you...
Well, I would just say really quickly that it makes me think of something that you said in the talk about a sort of a killing ceremony or a killing. So one of the differences that seems to me that makes it more okay for artists to do these kinds of things is that there's an endpoint to the project, right? And that the living thing or the technology is controlled in a way because it has that endpoint, or because you're not germline engineering, you're not developing a technology that you want to sort of release into a clinical setting or into a long-term research program, but rather it's sort of an episodic kind of event or whatever, right? And so it seems to me in that way, artists don't have, the issues that the scientists do.
And in the case that I was talking about, that clearly made the art less important.
Oh, I see.
So yeah, I totally get what you're saying, and this is kind of an interesting, it's a double-edged sword. I think I really like the idea of art being perceived to be frivolous because it generates two really interesting results. One is the fact that when we engage with those issues, we have no utility to hide behind. Yeah? So we can't just say, we have no time to talk about it because we're trying to cure cancer, or we have no time to talk about it because we're trying to feed the world or whatever. And this is often the response that you would get. I met a scientist who was trying to develop a better breed of yeast to make better beer, but when he was confronted, he said, I'm curing cancer, or, we're scientists, we cure cancer. So it's always kind of this utility is something that seems to make it more important, while in a sense what makes art important is exactly the lack of utility. So I was speaking to Martin Kemp, the art historian, and he was talking about the fact that art doesn't have a utility, but it has a very important function. Yeah, so there's a difference between those things. But with your story, it reminded me that we had a show which involved the killing of a goldfish. It was a collaboration with a neuroscientist. Before we started to grow nerve cells over electrode arrays, we used to use part of brains of goldfish to drive robots. And we are a group of about 6 or 7 artists working on this project and one scientist. And some of the artists refused to be involved in this project unless the scientist would publish a paper. And the scientist stood up and said, if I'm going to publish a paper, maybe 2 other people are going to read it. It's not going to make a dent in the world. If you're making it into an artwork, and we did it in a big festival, at least 50,000 people are going to be exposed to it, 50,000 people are going to think about it. It's way more important than my meager scientific paper that no one's going to read. But some of those artists said, this is the only way we're willing to do it. We want to feel that there's some utility, even if it's perceived utility, to give us a license to engage in a killing of a goldfish. Yeah. Which is, and those people eat meat, yeah, which I don't really get. Yeah, so.
You've touched on this already a little bit, but I would like to know, you mentioned Mary Shelley's Frankenstein a few times, and what the audience's reaction and perceptions are in relation to mad scientist or the societal perception of that, because your audience reaction to that would be vastly different than what a scientific paper would be in terms of mad scientist perception.
It is a really interesting issue, and it's very culturally dependent as well. And there's often, this is when Boris was talking about scientific kind of contracts, I'm often being referred to as being a scientist because I work with the tools of science, which is kind of an interesting thing. It's like you wouldn't call a painter that mixes paints a chemist, or you wouldn't call a photographer, a scientist, but there's still a perception. But because we're using instruments that at the moment are being kind of used within scientific context, that makes us scientists automatically just by using those tools. Yeah? So we often, that's one of the worries that I hear. UK is a really interesting place. So in the United Kingdom, scientists are extremely, there was a paper that was published in the journal Nature a few years back, which was titled, "This is the one case where you can't confuse scientists with facts." And it was the story about, they compared the public perception of science and then compared the way scientists think the public perception about them is. And across the board, scientists were highly respected, and across the board, scientists thought that they're totally not respected. Yeah. So it's kind of an interesting situation where they've got a huge chip on their shoulder and they're always concerned about that. But then the other thing is kind of the interesting thing is like, I don't need it to do this job. The vacanties that presented the mouse and the Urensbeck without preparation, this poor geneticist in Israel that is the naked chicken was showing, you know, those kind of things are generating those conversations. What we're trying to do, we're responding to them, but often, more often than not, there is an issue of killing the messenger. It's way easier to kill the messenger than the perpetrator. But, you know, I think one of the most beautiful lessons from Frankenstein is this issue of responsibility. And I think one of those of you who had the pleasure of reading it, because, you know, we tend, it's in our public imagination, but very few people actually read the original book. It's the creature who's this rational being, and Victor, the scientist, is the one who can't deal with the extent of what he did. And the creature becomes a monster when the scientist rejects him. So what we need to do is also be responsible for what we're doing, not just send it out into the world and hope it would thrive, actually be there. So this idea of responsibility and care and hospitality, which are part of the discourse within kind of new philosophical ideas, but not that new, you know, so within new materialism and feminist discourse, those things exist. We need to learn from that. It's like, I feel really bad actually sitting here as a middle-class, middle-aged white man and trying to tell you how to think, because it's really not up to me. Yeah, we need other voices, and that's another really important thing. Other voices that would actually help us direct where we're going and not leave it to the middle-aged white man.
So, Oron mentioned the reaction of politicians to certain ideas from science, and you know, it's an interesting question, right? The willingness of legislators to fund science depends to a certain extent on a certain degree of hype about what's possible, and our goals sometimes fall short. But you know, in the United States, we continue to invest tens of billions of dollars for civilian research, and even more on for military research that might be even past some of the cutting-edge things that you're trying to work on. But the other thing I was thinking about in terms of the responses is, in mad scientists, is that puts even more pressure on our education system with respect to science and the ability of the broader public to evaluate scientific claims or to understand scientific practices. Today at lunch, somebody from the Humphrey School used the scientific method as a term, and in science education, they don't use the term anymore. We talk about scientific practices that people engage in, and so we need a broader array of people prepared for understanding what those practices are and the results that might be reasonably expected from those scientific practices compared to some of the more outrageous claims that people are making.
And I think along those lines, it's an excellent point, is education, early education and adapted education. So that it's not, you know, the biology that we had 30 years ago, and that it's being updated so that when these new issues, technologies come along, there's a basis for dialogue. I think it's critical.
Yeah, and speaking about critical, I think critical thinking is one of the most important tools. This is, you know, but the problem here as well is how it's being cohered. So this idea of skeptic, it's not a climate change denier, yeah? It's someone who can actually understand the facts and engage with them critically rather than kind of the way it's now being exploited.
Yeah, and I was just going back to Boris's initial sort of prompt about the interdisciplinarity. I mean, for me as a historian, this is one way to get students to think differently about these kinds of technologies. So if in the news today, it's the case that some researchers in China are claiming that they've genetically engineered some human embryos that have been successfully born, and they've knocked out a particular gene to protect these twins against infection from HIV. If you draw people back through history to these various kinds of claims that have been made and these various, with new technologies, with scientific challenges that are real and you sort of walk them through the response and the ways in which we've tried to address these things, I think it helps to take some of the air out of the immediate sort of freak out response, and to sort of critically assess, okay, what really is going on here in terms of the biology, what's going on here in terms of hype and media and these other kinds of forces that influence how we get this information in the first place and then how we respond to it.
And saying that, here in America you have the liberal arts, which we don't have in Australia, for example. This idea, this concept that actually college is a place where you actually build a foundation rather than specialization.
We used to have it.
Not anymore, all right, sorry. Feeling bad for you then?
We used to have it in Wisconsin.
So there's foundation there that can be built on. But I must admit, I spent last night flicking between Fox News and CNBC, and the world you live in is amazing. It's like, how can't you exercise critical thinking just by flicking one channel back and forth? It's like the most amazing two narratives that are constantly opposing each other, and you have to understand the world is somewhere in between, and you have to be critical of what you experience. I think it's the best exercise in critical thinking. Just do that one evening and never sleep again.
Okay, yeah, so I'm still grappling with the thing you prefaced with talking about 19th century institutions, 20th century mentalities, 21st century technology. I was wondering, firstly, can you deconstruct that a little more simplistically for a 21-year-old? And then secondly, as someone like yourself who, given the last hour, I can safely assume has a rich depth of biological knowledge, I'm curious as to why you use the term white man.
All right. Good question. See, I think you are the future in a sense. First of all, you were born into the 21st century. Your life is still controlled by old farts like ourselves, but actually you are the people who actually need to engage with some of the mess that we're going to leave behind, and I think you're doing a really great job. I have a 14-year-old girl that I'm amazed by how sophisticated she is. So I think, but it's going to be a difficult process until your generation would actually be able to run the show. And then I don't think there'll be this idea of a middle-aged white man won't be that much of an issue. At this stage, especially in places like Australia and here and in other places, it stands for those 20th century mindsets that are driving the 21st century technologies. Yeah? So that's one thing. To do with 19th century institutions, as Harari was talking about, so many of our institutions are based from the hangover, because when the Industrial Revolution happened, and we can compare our time now to the time of the Industrial Revolution, and actually I think Mark can do a way better job than I do in kind of describing the collective trauma that humanity's experienced with the introduction of the steam engine, but generated then those institutions were set in place, rejecting what happened before. So we are now in a very similar situation. So things like universities, museums, as Harari says, our political systems are all based on a response to the trauma of the Industrial Revolution, but are not capable of dealing with the new trauma that we're bringing about through things like biotechnology and artificial intelligence that are going to drive a lot of the way things are being done in the world. Yeah? So the problem is that I would like to preface, the idea is actually, I think the technologies we engage now are way more powerful than the 19th century technologies and way more powerful than the 20th century technologies, but because the decisions we are going to make now are going to have a much more cascading impact on the future than whatever happened before. This is my kind of sense, that if we're starting to manipulate human genomes, germ cells, if we're starting to create artificial intelligent agencies that are actually stronger than us, if we're starting to create bureaucratical systems that are totally inhuman, if bureaucracy was inhuman up to now, it's going to be way worse in the future, when the outliers might not be able to engage in the world because they're different. Your generation, which actually embrace difference, would be the one that will have to break those walls and fight that.
So I'll try a couple examples from a non-scientist, non-artist perspective. So one of the institutional review boards are now common at universities in an attempt to create an institution to respond to the ethical challenges presented by a variety of things. Oron mentioned that he's operating a level 2 biological laboratory, the whole notion of different degrees of protection in biological laboratories is a institutional response to the risks of certain kinds of research, particularly with pathogens. So in a way, I would disagree to a certain extent with Harari's notion that we haven't, I totally agree, we haven't caught up completely, but there are places where we have managed to put in place some responses to the new technology, even though they're incomplete and they're just not transparent to people. For example, in addition to creating these laboratories, we also have a system of surveillance of those laboratories and sort of testing the degree to which they are actually protecting the way they're supposed to. And those all operate behind the scenes in a way that the general public does not perceive. And to the extent that Oron brings some of this out in public, then I think that's a gain to public understanding of some of these new challenges and some of the institutions that are in place.
But let me also just kind of I started by saying we have no cultural language to engage with it. If we have no language to describe what we're dealing with, we won't be able to make decisions about it as well. So I think the first thing we need to do is also start to develop a shared language that we can actually engage. And that's where the transdisciplinarity is really important, because when I talk to a scientist, when a scientist talks to an engineer, sometimes they're using the same words but mean very, very different things. So we need to figure out also ways in which we can develop shared languages and across the board. I would also like to see, it's really nice to see, but obviously here in America, it seems that the pre-actionary as opposed to the precautionary principle is taking over. So the difference is in Europe, they use the precautionary principle where basically technology is not released until it's proven safe. Here, technology is not stopped until it's proven out unsafe. Yeah? So you have a very, very different approaches. And there's good, there's both sides to those things, but we have to get more and more concerned as those technologies are becoming more and more powerful. And the most important question is in the hands of whom those technologies are sitting.
Can you sort of follow up on what technologies you're thinking of that have been released prematurely in the US as opposed to overseas?
So I think one of the really interesting things that we have, and that might not be even, it's going to do with mindsets. Yeah? So when I talk about genetic engineering or GMOs, I have no problem with that. I don't think that they're unsafe. But the way they were released here in America, the way companies like Monsanto capitalized on that, the way it really changed cultures all over the world, agricultural cultures all over the world. They might have fed the world, but there's a debate on how much contribution genetically engineered crops actually added to food production as opposed to how much more profit for Monsanto they generated. There are issues about that. And the fact that there was no debate here in the United States, while in Europe, I would argue there was too much debate as well. So they blocked things which are extremely useful, like the golden rice, for example, or others, because of their concerns around it. So that's why I'm talking about the fact that there's two sides to the story. But I think that we always need to be concerned about the cultural and societal perceptions and engagement with them. That's why I was so happy when 3 people spat out the steak that we served them. The fact is, we don't need to swallow everything we're being fed.
Just an observation. You guys sort of keep me thinking. The question and the issue we're dealing here is not just life. And yes, it seems that we don't have enough language for that, but it's also the issue of death. And it seems that this is at least as pertinent as life, because even those images that you've been showing, what they bring up the insides of our bodies. And still, no matter how advanced our sciences are, we still understand the insides of our bodies, or we perceive the insides of our bodies as death. We still have a problem with that. We still have a problem with being inside out, or even just shown such minor, because in comparison to what you can see on the TV, this is quite minor. But psychologically, we still have a problem with what is inside us. So it's just an observation. I sort of don't know what to do with it. But.
So Mark and I started a conversation yesterday about a social psychologist named Jonathan Haidt, who wrote a book called The Righteous Mind, in which he argued that human beings have evolved some moral foundations that underlie our intuitive response to certain things. And so blood, gore, and those kinds of things could be part of that evolutionarily conditioned response to avoid those kinds of internal, because they represent risk and initiate a sort of flight or a question of purity, that infection, you know, as we developed instinctive responses to bad smells and other kinds of things as ways of avoiding infection. And so one of the questions is, to what extent can we overcome some of those instinctive reactions or intuitive reactions and engage other neural processes so that we can take other, you know, react in a different psychological way?
Do you have the gag laws here in the state, where you're not allowed to show slaughter of animals and stuff like that? Is that because some states here have, yeah, in some states you have it, with this idea of, you know, you don't want to harm the prospects of the meat industry from selling their products, yeah? And I think it's really interesting in Western societies, and I try to engage also with non-Western societies and their way of thinking about life, and it is really interesting to see how this idea of real death, yeah, so there's the simulated death that people here experience on a crazy amount, but real death is not something that you are exposed to, while many other cultures, it's part of the culture. I grew up in a farm, for example. I've been exposed to death constantly. I've seen animals being killed, I've seen other things being killed, you know, I was involved with it. My relationship is very, very different than people who grew up in the city, and from their perspective, you'll never, obviously, call a beef a cow. You would, you know, this is the abstract thing that the meatpacking people were able to disassemble for you, and there's no connection between that product in the supermarket and the actual original animal from which it was taken. So I think what we need to do is also somehow open up those conversations. I think it's really important for people to see the inside, not just of ourselves, but of other animals. We live in this amazing time of those cognitive dissonance that we need to exercise constantly. So I talk about my ethical guidelines is to live a life based on informed hypocrisy. I know I'm a hypocrite, but at least I'm informed about it. What I can't deal with is righteous hypocrites. Yeah? And somehow, if we can do that, we can at least strive for something. We'll never reach it. Life is based on violence, but we need to choose, and we have to be aware of the amount of violence that we're willing to exercise towards the other, not whether we are going to exercise violence or not.
We do have really strict rules about what animals you're allowed to see slaughtered. I think there's only one meat processing company in the state of Minnesota that you're allowed to tour, and the rest of them, like, you will get arrested if you try to ask them to see what they're doing. It's Lawrence Meats in Cannon Falls. Go there, it's super cool.
It's a bit.
I thought of this when you were talking about different cultures. Your work has been displayed in different parts of the world, and what about the response to it in different parts of the world has surprised you? And yeah, that's my question.
Yeah. No, it is, and it is something I'm trying to plot, but obviously not in a scientific or sociological way, but just by experience, so it's going to be totally anecdotal. But what's really interesting, so here in America, it's actually really nice to have this conversation with you, and I think the whole setup actually allows for a deep conversation where usually I get very utilitarian questions and how you monetize the kind of work I'm doing. When I talk in Germany, the ethical questions are just kind of bubbling up, and there's obviously, they've got a very heavy weight of history that they still need to deal with, and they respond in a very emotional way to our work. When I talk in places like Japan, it's interesting because Japanese are more than happy to see technology becoming life. They have a major issue with life being manipulated, even though they exercise extreme violence towards plants with their bonsai. They still have to kind of separate those things. China was a really interesting place where they were talking to me also about this idea of harmony of nature and the fact that you need to somehow separate it from art, and art is about depicting nature and not engaging with it in an instrumentalized way. So they would see my work as instrumentalizing nature in some way. Nature is an extremely problematic term anyway. Where else? Yeah, those are the main places. Then, yeah, in different places in Europe, but there's also very, very different contexts. So when one of the works that I didn't have time to talk about was trying to put natural history museum specimens within an art gallery, encased within luxury display cases that we commissioned a luxury designer, kind of a luxury good designer. So the idea there was to confront the audience into whatever, because you enter a gallery with a specific contract, you want to see art that was curated by people like Boris, who he knows what art is, and he's the gatekeeper. Yeah, and if it's in, it's art. If it's out, it's not. But then they see those natural history museum specimens, like, are we in a natural history museum? But then they presented as object of desire, and you just want to pull out your wallet and buy them. So the question is really about those institutional contracts. So when we show our work, for example, and we did a mistake of showing my work in like straight science museum. So when we presented our Victor Mesleder in the, whatever is the name of the science museum in Ottawa, it was only about illustrative of scientific processes. There was no place for you to have a deep discourse into kind of the ethical issues or all of the other issues that we are trying to raise. When we showed it in MoMA within the design context, as I said, most of the readings that we started to get feedback was, you're not really good designers, you failed. Yeah. So it's really context dependent as well, not just culture dependent. Yeah.
You, to follow up on that, you mentioned the location of Perth in Indigenous land in Australia. Have you confronted Indigenous people in Australia with this work, and if so, what kind of reaction do you get? And sort of related to that, when you did the death ritual, which traditions did you draw on for the ritual?
Yeah, that's a great question. So first of all, I just put in context, Australian Indigenous people went through a cultural and physical genocide. There's only 1% of the population is Indigenous, so you don't really engage a lot with them, but we are trying. We're actually trying to work closer with the Center for Indigenous Studies at our university. One of the main keynotes in the conference we organized was a young Indigenous science fiction writer and a lawyer, and she told a really beautiful story about kind of how she built the trust with us, and this is the whole idea of building trust. We had a couple of Indigenous residents, actually, and one of our master students was Indigenous. You know, it's not a unified thing. The Australian Indigenous people there's about 350 language groups. They're more diverse than Europeans, you know, so clustering all of them. But they do have a very strong relationship to animals and others, so we have issues with some of our Indigenous visitors and residents that there's some animals they're not allowed to come near to. And there's some animals they're allowed to engage, so there's lots of protocols around that. When it comes to the protocols of the killing rituals, we didn't really, you know, that's kind of the interesting, there's kind of a bit of a letdown because we invite people to the killing ritual, but then we say, why are you here? You know, you killed more cells when you brushed your teeth this morning. Why those cells are more important? Yeah. So we have those, but we had, when we had a big show in Dublin, we had an Irish wake, which was nice because it involves lots of drinking as well and having the dead thing in front of you while you're having a good time. Yeah, so there's different ways of dealing with it.
More questions?
If somebody else wants to. I guess another thing I was kind of curious about was pushback from, have you received actual pushback from religious groups, ethical groups that were actually, that somehow succeeded in interfering with anything you were doing or caused any kind of trouble?
No, not really. It's kind of interesting that the main pushback we get is from institutions that predict a pushback. So, which is really interesting. So museums and galleries, they just don't know how to deal with it, and they would go, you know, and I suppose they need to do the risk management plan, and they have to come up with the worst case scenarios, and they freak out, you know, once they start to think about it. When we had a show in France, where actually we had a show where we ate a steak, It was one of the biggest art and biology shows in Europe. It was 2003. There was a group of protesters, the mob of Juget. I don't know if you know the French people that would burn McDonald's and would rip down GMO crops. And so they were there. They were ready to burn down the gallery. And the curator did something very, very smart. He basically he did the opening outside of the gallery. Those people were around. He invited them to come and see the show before they make a decision. They came and saw the show, and they shook our hands and said, "You're doing an interesting work. It's important. It's important for us to be exposed to it. It's important for other people. It's important to think about it. We don't want to burn you down this time." So it was nice. We had even one gallery that employed a PR company that said, "We need to engineer a protest against your work to generate noise." We don't want that. So no. So it's actually, I think, once people are exposed to it, once people are, this is the idea, that we are engaged in those places where we have no language to describe. And I think when people are exposed to that, they realize that we challenge them to think about it, but we don't tell them how to think about it. And this is, I think, the power of art as well, this ability to be ambiguous but challenging rather than be, and this is, there's some American artists, and I can understand the situation here, that sometimes you need to shout very, very loud in order for you to be heard, and sometimes there's very important issues that you feel that you need to be very didactic about it. I think those are really great pieces of propaganda. I don't know about the artistic value of them.
Should we just wrap up? Okay, should we wrap up? Any more questions? No? Oh, there is one.
Oron, thanks for your inspiring talk. As a person who was trained to be a scientist, I saw you and I was very inspired that you were able to travel between this medium of art and science, and this is a realm that's very difficult to define. And how I see the education system now is everyone's trained to be in a very specialized field. Either someone is a scientist or someone's an artist. But I think what you brought up a point, which is this interdisciplinary thinking, is very important. So if you could provide a curriculum to forge that thinking, how would you do to promote that?
I would get someone like Boris and pay him really well and get him to engage. And I think you're very, very lucky to have someone like Boris here and have people who are willing to engage with that. So this is really inspirational. And coming here was extremely inspirational. And I would add another thing. Those revolutions would start from the margins and not from the centers. New York is boring. London is boring. Those people are parochial and inward-looking. I think those places like Perth and Minneapolis and Dublin and those places in the periphery is where those interesting things are happening, because those are the places that allow this cross-thinking that is so important, as you mentioned. So first of all, I would like to congratulate you for having something like that here. So this is great, and you should cherish it, and you should try and help Boris as much as you can, because... yeah. Okay.
Thank you, Oron. But I will throw my 2 cents into that question. I think we should be well past multidisciplinarity. We should be well past thinking about disciplines at all. We should deal with issues, we should deal with cultures. The situation where we have, again, 5 different professions on a stage to understand something indicates that the way things are understood and studied is extremely ineffective. And the way that we need to do it in the art museum, because there are not that many other places who can do that, we should go past disciplines, past multidisciplinarities, and just think in completely different terms. I want to imagine a department where, again, Oron and Branda and everybody else on this stage actually work together to figure something out. I don't know, maybe with that we'll just really wrap up. Thank you very much for being here.